Hello, I am Mary Ellen Geiger, Chair of the AAPM Data Science Committee, and along with me is Paul Kinahan, Chair of the AAPM Research Committee. We are the two AAPM Principal Investigators for MIDRC. The COVID-19 pandemic presented an urgent and critical public health crisis. Biomedical research was needed to address surveillance and early detection of COVID, differential diagnosis and prognosis on severity of the disease, predicting and assessing response to treatment, and monitoring the post-COVID patient. The question is, what is the role for imaging and AI in addressing those needs? Here on this slide, I show various chest radiographs and CT scans of COVID at various stages. During the past few years of the pandemic, many AI methods were developed and published. However, none have reached clinical practice in the analysis of medical images of COVID. Why? Well, perhaps there are poor quality of data in these Frankenstein data sets with mislabeled data and multiple unknown sources. Also, the existence of duplicate data where there's leakage between training and testing. Perhaps a lack of communication between the AI experts and the medical imagers and medical physicist experts, with lack of valid ground truth or incomplete understanding of how to test these systems. And also biases. Perhaps the data were collected for a specific clinical task or a specific population. The medical imaging community response to the COVID-19 pandemic resulted in MIDRC, the Medical Imaging and Data Resource Center. It is led by the AAPM, the ACR, and the RSNA. It was established in August of 2020 and is supported by the NIBIB and is hosted at the University of Chicago. MIDRC is a curated image and data commons, enabling researchers to address topics no single image archive could yield independently. It is a collaboration of over 24 institutions and 100 investigators from academia, community practices, FDA, and others. The two scientific components of MIDRC are: one, an open discovery data commons for creation, testing, quality assurance, and data connectivity, and machine intelligent computational capabilities by developing clinical relevant algorithms and software tools. Data is collected by MIDRC from various medical centers and hospitals. Currently, we are mainly taking in chest radiographs and thoracic CTs. These come into MIDRC through two input portals, one from the RSNA and one from the ACR. Once in MIDRC, these de-identified data undergo curation, annotation, quality assessment, and other processes. These are then made available at the output portal on Gen 3, which is data.midrc.org, for various AI developers for cohort selection, downloading of images, and others. As of June 22nd of 2022, we had ingested over 124,000 imaging studies, of which 92,000 are undergoing curation, with 31,000 currently released to the public. The infrastructure of MIDRC was built through 5 technology development projects. These are led by the various organizations, and note that number 3, TDP3, on developing and implementing quality assurance and evaluation procedures for usage across MIDRC is led by AAPM. This is further developed using 4 MIDRC data science subcommittees. One on data standards and information technology, another on data policy and procedures, another on data quality and harmonization, which is led by AAPM, and another on data search and user portals.
We will now review some of the unique attributes of the MIDRC image data sets. One of the unique and important attributes of the MIDRC data collection is the curation of the DICOM images and the associated metadata. On the left is a sample of DICOM images that came from a chest X-rays cohort. You can see the wide variety of orientations, exposure, and image quality. This illustrates the variety that occurs in standard of care imaging. This variety is normal for clinical imaging, but could confound algorithms that are expecting uniform images. In addition, towards the center there is an image of knees due to a mislabeling of the image when it was acquired. Variations and frank errors in DICOM image labeling do occur in standard of care radiology imaging. A goal of MIDRC is to provide information on the quality of the images and to allow for the selection of patient images cohorts of a known quality. The process we use is shown on the right, where the DICOM images, indicated by the blue cubes, start from the contributing sites, are de-identified, ideally using a common protocol, but not always, transmitted to either the RSNA or ACR pipelines, where they are reviewed and further de-identified if needed. A process of image annotation is under development. An important point is that the DICOM images are not modified or corrected, but rather we collect information about the image. This information is abstracted to a database maintained by Gen 3, which is used for patient cohort selection, which I will describe 2 slides from now. The study description identification occurs at the step indicated by the red arrow where the LOINC codes are introduced. A critical element in this is determining the metadata, such as body part or study description, when it's incorrect or missing. Perhaps surprisingly, there is no uniformly implemented process for radiology study descriptions. Recognizing this, the RSNA RADLEX, short for Radiology Lexicon, group several years ago started the RADLEX playbook. In parallel, the LOINC group, short for Logical Observation Identifiers, Names, and Codes, which was initiated by the Regenstrief Institute, was expanding its efforts to introduce common terminology for laboratory and clinical observations. The 2 groups partnered to develop the LOINC RADLEX playbook, which provides a unique set of study descriptions, body regions, and other essential descriptors. These are identified by a unique LOINC code. The LOINC database is recognized by the National Library of Medicine and is part of the medical metathoris. The MIDRC process reviews the DICOM metadata and image and assigns a LOINC code to each study to facilitate accurate cohort selection. Using the information about the images collected in the database, the Gen 3 Data Explorer can now be used by investigators to select the types of images they want to download. This includes selecting the imaging modality, such as CT or X-ray, the body region, gender, disease status, and many other parameters. An illustration of the Data Explorer is shown on the right. The Data Explorer is accessible from the MIDRC website and directly from data.midrc.org. The data dashboard, shown on the right, is a live representation of the available studies, and at the end of June 2022, there were almost 12,000 patients with 32,000 exams or studies and almost 100,000 imaging series as shown listed by modality.
Another unique aspect of MIDRC is its aim to collect diverse and representative data. As shown here at the bottom is a pie chart of the distribution of different races of data collected in MIDRC. This is of June 2022. We aim to do this by increasing where we obtain contributions. Currently, contributions are coming from 23 states, where imaging data is in the pipeline, ranging from completing data contribution agreements to ingestion to curation and then to release. Another unique aspect of MIDRC is that it includes a sequestered data commons. Here we have in the green a open data commons. So 80% of the data coming into MIDRC is publicly available for research and development of algorithms. However, roughly 20% is sequestered. It's restricted from public access, and it is for use with finalized algorithms, not those under development. This is to accelerate regulatory clearance of AI algorithms and act as a large test set from which task-based samples can be drawn to yield real-world performance on representative data. For performance evaluation using the sequestered data test sets, test cases will be drawn from the sequestered set according to the distributions related to the specific task, such as the clinical task, the claim, and the population. That is, the sequestered set in the entirety will not be used in the test, thus maintaining the integrity of the test set. Next, for a specific task, a sample set will be drawn from the distribution for use in the testing. Additional tests for the same task will require a new sample set, as shown here. MIDRC also funds various research projects as shown here. Of these 12, CRP 9, 10, 11, 12 are under the AAPM umbrella. Collaboration among these projects is further facilitated with 3 trans-MIDRC scientific work groups. One on the grand challenges to create and coordinate efforts on all aspects of challenges, another on the bias and diversity work group and the goal of assessing and mitigating bias in data and machine learning, and another one on labeling and annotation. To further see the accomplishments of MIDRC, we have here MIDRC by the numbers. You can see we have various imaging studies ingested and released. We have various publications and presentations, as well as algorithms created through MIDRC. We have 184 registered users, over 100 investigators, and over 100 collaborating institutions. To further benefit the research, interoperability efforts are essential. Here, MIDRC is working with N3C on acute COVID EHR data. We're biodata catalysts with the PEDAL data for clinical and multi-omics data. Here, with mutually beneficial enrichment of data sets with information across scales, pan, and multi-omics, we will improve the understanding of COVID. We are also undergoing federated learning in AI through a collaboration with Argonne on privacy preserving federated learning in distributed enclave. MIDRC is a community. It has a monthly seminar series highlighting the work of our researchers. It has quarterly town hall meetings as a forum to engage directly with the medical community at large and answer all their research and contribution questions. Please check out our YouTube channel, MIDRC Media, for recording of past events. MIDRC has an interactive decision tree for recommending performance metrics for use by AI developers. So please, for more information, visit midrc.org or subscribe to our newsletter for updates and upcoming events. MIDRC is a national resource. It aims to support the medical AI ecosystem with curated, representative, trustworthy data. It assesses diversity and aims to mitigate bias. It's a community with the culture of collaboration. And because it is open, it is lowering the barrier of access. And in order to accelerate translation of AI, real-world quantification of algorithm performance will be conducted with the Sequestered dataset. We will soon be entering year 3, and the goals then will be to look at long COVID, beyond lung to include cardiac and brain, and beyond chest radiographs and CT to include MR and ultrasound. We will be also expanding our Sequestered data commons for translation and furthering our interoperability efforts. And then, what's next? Beyond COVID, other diseases and conditions from other funding, though. So note, for a given medical imaging, a patient has already benefited through medical care. A hospital and medical center has already benefited through reimbursement. Now the public can benefit with the MIDRC secondary usage of those images. We can all help change the future of medical imaging and increase its impact on public health. Thank you. And for more information, please come back to booth 1085 in the exhibit hall. Thank you.